that the organization structure has evolved. And really, we have a steering committee. You guys meet every 2 weeks. We call it something else, but it's really the steering committee meeting. I want to focus on the 3 arms that come off the administrative core. We have a study coordinator, David, who's done a fantastic job working with all you site coordinators. You can see on the left, we have 3 research projects, scientific cores. Again, this has evolved. We didn't originally have a data management core. We have that David is leading. We have a protein and DNA lab core that I will talk about in a second. So this is the organizational structure, and our vision for SickleGenAfrica is to align the survival of individuals who have sickle cell disease in Africa with national norms. And the reason why we say that is because the life expectancy of sickle cell patients is markedly lower compared to those who don't have sickle cell disease. And organ damage, end organ damage, that is due in part to the disease process I described, contributes to that. And so our vision is that we will have enough knowledge to be able to intervene so that a sickle cell patient born in, Timbuktu will live to the same age as somebody who doesn't have sickle cell disease. To do that, we need to develop prognostic biomarkers of organ damage through the discovery of key genetic modifying factors and cognate mechanisms. And so the major causes of death to your right in cardiovascular, respiratory, and renal diseases. And so the overall scientific premise of SickleGenAfrica is that these complications that cause disease are driven to a large extent by hemolysis through inflammation, oxidative stress, and altered nitric oxide biology that we believe impacts the health of major organ systems, which in turn lead to these cardiovascular, respiratory, and renal complications. And I've already explained to you in a couple of slides back that we have cytoprotective proteins, haptoglobin, hemopexin, alpha-1 microglobulin, phemoxinase 1, that normally will shield us from the deleterious effect of hemolysis. But how much anybody makes of these proteins is influenced by their genes. And so the genetics of how much of these proteins and the overall cytoprotected phenotype is really at the core and forms the scientific premise of the study. So our overall hypothesis is that genetic variation influences the body's defense against hemolysis and the development of organ damage in sickle cell disease. And to address this, we came together as a team. We started this process probably about 2 years ago now when we wrote the grant and put together a collaborative team. Pittsburgh originally was going to be the bioinformatics core. There is the opportunity for that to still happen. The person who was leading that has left. We've identified somebody else who has shown a lot of interest, and so the bioinformatics core may still happen in Pittsburgh. The good news is we don't really need that yet, because it's the kind of stuff we'll need perhaps towards year 4 and 5. Ghana, where I'm speaking to you from, is where the administrative core is based. The molecular and hematology and sickle mouse core is going to be running soon. We have a lab set up. I'll show you a couple of photographs of our first equipment. Project 1 then is the Genomics of Hemolysis Modifiers, and that's the project that I lead. It's essentially a GWAS. In Tanzania, we have Judy Makani, that some of you just heard speak, leading the malaria project. And then Professor Sani, in Kano, he's a cardiologist, leads project 3. We work very closely with H3 Bionet that is based in South Africa. Nigeria also has the biorepository core in Abuja, and I will show you some pictures of some of the activities from that core. So our study population, we are studying overall, we are hoping to enroll 9,000 sickle cell patients. You can see in the right, the cities and the target for each of the clinics. I don't have to go through that. You can see the numbers yourself, just to save time. The strategy then is to consent. You can see it says 7,000 here. That's fine. We originally were thinking of 7,000, but especially in Nigeria, in Lagos, where we were originally planning to recruit only adults, the pediatricians are very excited and they are participating. And so we've upped our numbers. This is an older slide. And so overall, we're going to study from a phenotype point of view, the malaria, whether people have malaria, the degree to which the malaria is severe, whether they have hemolytic anemia, lung function. We haven't started a spirometry, but this is something that we'll do. renal function we'll do from the samples we get. And as I mentioned, the echocardiovascular phenotyping, I'm pleased to say, I think just started in Kano a couple of weeks ago. The design is basically a case control design of genome-wide association studies. We're going to use extreme phenotypes. So for each of those markers that I mentioned, the plasma proteins, we're going to study as cases individuals with the very lowest level, that's less than 25th percentile, and individuals with the very highest level. And if we use this case control, we'll be able to identify genetic signatures that determine the level of these proteins. We had a fantastic launch less than a year ago, and it was very nice to see all of you, my friends here in Accra, for us to launch this. And I'm most impressed and actually very happy that we didn't just do an Accra event. We basically did a rolling launch. And so we kicked it off in Accra. Then we went to Kumasi, Abuja, Kano, Lagos, Dar es Salaam, all within about 8 days to launch SickleGenAfrica and have at the back of that a community engagement focus group that really was very, very helpful. So what you're seeing here are some of the pictures from the inaugural ceremony that we held at the La Palm Hotel here in Accra. As I said, we didn't just do a launch, have speeches and go home. We tagged onto that a community engagement workshop. And what you're seeing here are the dates when we visited these cities. And the focus group ideally consists of 20 participants, although in some cities there was a lot of enthusiasm. I remember when we got to Lagos, we had more than 20. In Kano, we had more than 20. But we tried to get representation from 6 different groups: adult patients, parents, government workers, and we really had a back and forth, which was very, very informative. Because keep in mind, this is a genetic study. And so what we wanted to do was to get buy-in from the local communities. Because after all, it's my personal belief that our genomes don't really belong to us. It belongs to our community. Our parents handed it over to us, and before them, their grandparents. And so when you're studying the genetics of any community, it's very important that you get buy-in from the larger community beyond the participants themselves. So Kofi Ani led our community engagement focus group, and this is what we went through. Again, for the interest of time, I'm not going to go through that. We are going to share these slides at the end of the talk, so everybody will, and this will be uploaded on the SickleGen website, so you can all get a chance to go and look through it at a later time when you have more free time on your hands. This is some data from the community engagement focus group. It's very, the last row is what I'm interested in. The proportion of targeted numbers. We had 100% in Accra, Dar es Salaam, Kumasi 95, Kano, Lagos not far behind. Abuja, we had 80% participation. But overall, this was very, very helpful. I think what follows are some pictures. You see some royalty here in the Accra focus group. It was very important for the patients to see that their local leaders, the local traditional leaders, were at this event, and that has really, I believe, helped us a great deal. The folks you see there are the local chiefs in where our hospital is, Colin Gono, and I don't know whether it has anything to do with it, but we are having a massive recruitment in this region, and I think it's because there is community engagement. So we did this in Accra the day after the launch, and then the following Monday, some of you went to Kumasi. These are pictures from the focus group in Kumasi. From Kumasi, Kofi Ani, assisted by Mary, a psychologist, went to Abuja. I joined them later on. These are pictures from Abuja. Kano, we all went very local. You can see me there in my full regalia there. It was a lot of fun. It was very hot. We were staring into a glaring sun. And so that's why you see that picture there. But it was a very, very productive community engagement activity in Kano. And then we moved on to Lagos, where again, I got to meet one of my heroes, Akinyanju, who is sitting in the middle there. This is a warrior of sickle cell disease. He's an old-time physician, still very committed, still very energetic, and it was very, very important to get his support. So the community engagement has been very, very, was very good, and the manuscripts reporting the outcome, some of which I've showed you, is currently in preparation. The final stop was Dar es Salaam, and what a beautiful picture showing people actively engaged. This is Frahini, who has left that since. I don't know whether she's on the call, but she was the site coordinator in Dar, and she did a great job pulling this together. So in terms of activities and our deliverables, community engagement, we can take that we've done very well to the extent that the community engagement focus group within H3 Africa actually looks to Kofi Eni and our work to model, which is quite, it's a nice feeling because we've only been at this for a year. They've been doing it for 6 years, but this had never been done by any of the networks. And Kofi is constantly consulted in how to spread this among the other network. Training, we haven't left training behind. The first H3 Africa meeting that we went to, I took along a cardiology fellow from Pittsburgh who is working on a specific aim 3 of Project 3. And here you see Maureen, who originally is from Kenya. She's become an expert in doing echocardiovascular phenotyping of sickle cell mice. And you can clearly see here, just looking at the left ventricle of sickle cell disease mouse is thickened just like it is in patients. And so what she's going to do is try and functionally validate whatever we find in humans in mice. In Ghana, we set up a Dean's Biomedical Research Internship Program, which was the very first of its kind in the University of Ghana, providing medical students the opportunity to do research during the long vacation rather than just, doing something else. And that was also very helpful. The Biorepository Core in Abuja recently completed training of a laboratory personnel from each of the 6 sites. So each site sent 1 lab technician. We sent a technician also from the administrative hub, and Alashli Abibiku, who is the core lead, and this is her lab, and what you see on the left are pictures of the lab technicians basically learning the proper way to process samples, do quality control, and also, most importantly, the proper way to ship samples to that biorepository. So we've had a plus from the biorepository core. The summer internship program I've already referred to, so I'm just going to skip this slide. All right. So the biorepository core has done well. The community engagement core has done well. The data management core has done extremely well. This is unusual, probably the first time for a webinar, but I think you should all give a round of applause to David Ajay, who has done fantastic. He's done fantastic. And what you're looking here so we're using the RedCap database, and this is just a snapshot a couple of days ago. This was a lot of work, as you all remember, in getting together the case report form. But now, we are using this. It's very, very good. I can sit at my laptop and see who is recruiting where. This is, again, a product of the data management core under David. Here you're looking at the adult electronic case report form. It's just a section. This is a section for the kids. This webinar has come at a very good time because we've just submitted our second media report to the NIH. And what we're able to tell the NIH is that as of January 11th, we had recruited 1,861 participants across all 6 sites. This is less than, basically, in some centers, 6 months. That, I think, is only done 6 months. Accra, we've probably done 8 months. It's a huge rate of recruitment that I'm very, very proud of. 57% are kids, 43% adults. If we break it down, as of 11 January, we had recruited 794 adults, with Accra leading the way, not surprisingly, as a very large clinic. You don't see Kano on this simply because Kano wanted to wait until they had the echocardiovascular machine before they did the adult recruitment, which they have started. With the kids, we had recruited over 1,000 kids, with Dar leading the way, which is impressive because Dar was actually the last site to start recruitment, and yet really is leading the way in recruiting kids. So in terms of our patient enrollment, we're doing very well. Project 1 is a GWAS project, and what we need to do is to measure the levels of the plasma proteins that I talked about and then do the genotyping. We haven't gotten to the genotyping yet. We'll probably do the genotyping in the 3rd year because we are going to use an Illumina chip. And my understanding is that there's a lot of variation from different lots of that chip. And so we'll wait till we have our DNA and we'll buy one lot of that chip and do the genotyping one time. But what you're looking at here is a capillary 2 flex piercing system. We did say that even though we are enrolling sickle cell subjects for our studies, we need to be sure that essentially that they have sickle cell disease. And so this machine, which was installed here in Accra yesterday, is going to help us confirm everybody's sickle cell status. We have another instrument from Siemens coming soon that will allow us to use an automation to determine the levels of the different proteins that I talked to you about. All right, so that actually was my last slide. So what I really, for those of you who missed some of the earlier slides, for me, the best slide really is the way the organization, in terms of the network, we've really held together. We've done a great job. The site coordinators, you've done absolutely fantastic. The first 2 years, the strategy all along, the first 2 years, we will aggressively recruit. And in fact, looking at the progress report, we've actually been operational about just 1 year because the money, we actually got the money, I think I have the date, 13th or 11th of January 2018. And so in actual fact, we've been operating for just 1 year. And to have close to 2,000 subjects recruited with plasma samples and Buffy codes stored is very, very impressive. Moving forward, we have to do exactly what we did last year, in the last 8 months or so when we're aggressively recruiting. We have to do the same thing again for another year and a bit, and then keep on storing the samples. The lab technicians, those of you who went to Abuja, please make sure you're storing samples according to how you were trained to do that. And then we in the protein and the DNA lab, we will start churning out data on the levels of these biomarkers. You all have the expertise, right? You all have the expertise now to extract DNA. We'll extract DNA. My plan is towards the end of maybe the 3rd year or the middle of the 3rd year, we start sending samples to the biorepository. We hope to get all the protein analysis done by the end of we're going to start now. And so in terms of timeline, we are doing great. And that's all due to everybody who's on this call and others who couldn't make the call. So I'm going to end there by telling you that the state of SickleGenAfrica is very strong and we're doing marvelous. We've become a model for other networks. And so thank you all for all your hard work.